Unit 1 is the chemistry of life. We have 8 units in AP Bio. So this one unit is just about 8 to 11% of the AP exam, which isn't too much. Some of the other units are more about 13 to 15%, but I really love this unit and I don't want to skip over it or have you rush through it because this is the foundation for the rest of the course. Okay. Also, just a little blurb there about the exam in general, encouraging you that we know that it's half a free response and half multiple choice. So that really kind of makes it a fair playing field for people who are either really good at the writing or really good at the multiple choice. Kind of makes it even for everyone. So in case your teachers hadn't already told you, that's the continued format of the exam. I put a list of Unit 1 vocabulary here as well for you to reference later. I'm obviously not going to go through each of these terms because your time is more important than that, but I did want to provide it for you as a resource. I like to encourage students that when they're looking at a list of vocab, don't just go to the book or go to Google. You've already learned this stuff, right? It is March. And so look at each of these words on your own and just have a blank piece of paper out and write down everything you know about that word, right? So macromolecule, boom, write down everything you can, right? And in those moments of forgetfulness, you'll realize where the holes are, right? So if you can think of everything, but then you realize, oh shoot, you know what? I actually don't know what builds a starch, or I don't know the difference between starch and cellulose. And that's the moment in your learning where you will go one step further than someone else who's maybe just copying down vocabulary words. All right, so topic number 1 is life and water. I made a little flowchart for you here to help you in your reasoning of if you were to get like a free response question and it asks you to describe, why, describe water's ability to have a certain property. And it's going to obviously want you to talk about polarity. And students get mixed up because they want to just jump right into polarity and property without talking about the structure of water. So the structure of water is important because it is what gives water its polarity. And I have a picture on the next slide for you as well. But the specific structure of water, right, we all know it's H2O, right? So we have 2 hydrogens and 1 oxygen. It has a very specific and unique structure. It's that structure that gives it its polarity. The polarity is then what gives it the ability to make hydrogen bonds or have hydrogen bonds with other molecules. And then that all together is what gives water its properties. Okay, so kind of reference this flowchart if you need to, but students tend to just want to jump right into the property and say what it is, like, oh, this is what cohesion is, and boom, I'm done. I answered the question. But you didn't talk about hydrogen bonds, you didn't talk about polarity, and you didn't talk about structure. So just kind of be mindful of the things you put on a page and that you're really, really describing things well and from a place of understanding, not from a place of memorizing. Okay, alrighty. So like I said, water is a polar molecule. If you think about polarity, think about, what they even talk about our nation, right? They say how like America has become very polarized, right? So it's very separate. There's it's the polarization of a magnet. You have a north and a south end. They are different from one another. So polar molecule has these regions of partial negativity and partial positivity. That's where that, oh, you know what? One second. I'm just going to make my captions a little larger. Okay. So these regions right here, that stands for partial negative. And here's our partial positive. It's not a full negative or full positive. It's because it's a covalent bond still, right? They're sharing those electrons. Electrons have not been displaced outside of the atom's orbit, but it's being held closer to the oxygen molecule. So here's another look at water where we've got those valence electrons here for oxygen. They're pulling hydrogen's electrons closer to it. And then one more picture that I want to make sure we clear up, that when we talk about the covalent bond in water, we're talking about the bond between oxygen and hydrogen of one water molecule. Where when you hear the word hydrogen bond, and you might be like, I know this, come on, like Mrs. Parnan, I know this, it's no problem. But for whatever reason, time and time again, students talk about hydrogen bonds and they're talking about the water molecule itself. and the hydrogen and oxygen, rather than hydrogen bonds are going to be between hydrogen molecules, right? So that's why you see hydrogen bonds a lot of the time represented by dotted lines, because your straight lines are representing a covalent bond. So those dotted lines or those dashed lines, they're not there by mistake. They're there to clearly show that, hey, this is an interaction, right? There's not like a physical bond that's happening. It's an interaction between something that's partially positive and partially negative. And remember, opposites attract, okay? If you have a positive charge and a negative charge, they want to be near each other. Or if you have like charges, they are going to repel. Thinking back to chemistry, why is water polar? The reason I want to show you the periodic table is that you might be given, and again, AP Bio is not about memorization, it is about application. So yeah, you can know everything you need to know about water, but if you can't apply what you know about water and its polarity and electronegativity to a different molecule, then you might be in a tough spot, okay? So for example, when we talk about electronegativity, that polarization that happens because of it, electronegativity has to do with an atom's ability to more aggressively pull electrons. That's how I like to think of it. I like to think of oxygen right here. Here we are, top right. Oxygen more aggressively pulls electrons toward it. Whereas if you think about like fluorine and chlorine and all those, we don't really see them doing that a lot because they're just more likely to form ionic bonds, right? Because they only need one extra electron. So why would they need to share an electron if they just need one more? So if for those of you who are ever wondering, like, why aren't we ever talking about, group 17 and 18? It's because they're not going to participate in those covalent bonds as frequently. All right, I'm just going to skip over to the chat, make sure we're all okay. Let's see here. Oh no, can people not hear me? Oh dear Lord. Some people can hear, hopefully. Let's see here. Looks like some people can hear. Oh no. Okay, some people can. Oh, okay, good. Oh my gosh. I was like, wow, I've just been talking to myself. That would be so embarrassing. Okay, good. All right. Well, I'm sorry for those of you who can't hear. Such a bummer because a lot of my slides don't have a ton of words. I just talk to the picture because actually, biology fact for you, our brains cannot process images, text, and audio, right? As smart as we all are, those three things happening at once is an explosion for your brain. Your brain will choose the one that it wants and only focus on that. So just kind of FYI, if you're ever going to be making a presentation, if you're going to give lots of words, let everyone take a second to read it and then chat away, because otherwise they're not listening to you. Okay, last picture, talking about polarity. It's just one more model, right? We talked about polarity with our partial positive and partial negatives. We talked about it in terms of electronegativity. Here's just another model where we're looking at why water is polar. Again, we all like to see it in different ways. I'll try to give you as many pictures as possible. But here, another thing to consider is that, remember, the nucleus of your atoms has positive protons on the inside. So those 8 positive protons are also playing a role in kind of attracting those negative electrons closer to the nucleus. Whereas the 1 proton for hydrogen, that's super strong. It's just 1 positive ion, so it's not going to have as great of an effect pulling those negative electrons to the nucleus. So if there's anything that you got out of the last 5 minutes or so, water is polar, right? Because oxygen is electronegative. It hogs those electrons, making oxygen partially negative, hydrogen's partially positive, and so we get this separation of charges, right, in a covalent bond. Okay? Because of that, make sure we're talking about water and things that like water or things that are polar, they're going to be hydrophilic as well. So when we start using words like hydrophilic or hydrophobic, this all goes back to polarity, right? Remember, it's not just like it likes water because it likes water. It likes water because it's polar too. And it's like, hey, you're polar, I'm polar, awesome. Like, let's hang out. Whereas if you're hydrophobic, you're not going to like water. And so you're not going to have separation of charges. You're going to have, maybe fully saturated atoms or stuff like that, where you're not interested in a polar water molecule as well. Again, I just talked about this before, but misconception alert. This is a pressure hydrogen bonds. It's interactions between molecules. They can cook so not within. So between meaning this is an application question, and I've been saying that word way too much. Wow, I'm like on the application within. Something. So you'd be talking about anyways, this is one of those questions where it seems like it's not related, but it's excellent. They're really just trying to get at your reasoning. We're moving right along. So these are also things that I'm just throwing out at you because I think you all know the properties of water well. It's more the application of them that you would need some help with, right? So this is something that you could look up in your notes and you have pressure, the atmosphere, water sticks to itself, and then these are your phase changes, the density, so like at what point of temperature, pressure, it floats on water, either ice or water or water. Not to say these aren't important things, but I know that you can either. I know that you know these. Think about feeling confident in that. So what we're going to do is look at examples water instead, right? To cook pasta. So number one, you probably done this lab, maybe even when you were a little kid, and you drop water onto a penny and you count how many. And again, elevation's not on this graph. And kids are always astonished. They're like, we're in the hundreds. This is incredible. Like the bubble grows and it's not falling. And I forgot. And that's a great example. More like a fun example, not biological. That shows water sticking to itself. This graph right here, and also something that could potentially be sticking to the penny. Right. So pressure. If we think about like a biological reason for that, this is something you need to make sure you understand. How pressure is. So if we're talking about how water goes from as you get higher into the atmosphere, huge tree roots. Like if I'm talking about looking at this graph, we should be able to redwood pressure decreases. I'm going roots from extend 1,000 really long down to 10 banks. So pressure is and water is picked up from those roots, brought all the way higher into the center of the tree. It's traveling up. So pressure is decreasing. I knew a fact for you of how it goes, but it's got to get up to the highest. Think about how that's going to affect. Right, so it's defying gravity, and it's because of water's cohesion with itself and adhesion to the xylem that it can reach to the top. Okay, I don't think you necessarily need to know what the xylem is, right? But you need to know that water is moving up some sort of a vessel, right? It's sticking to the sides of the vessel, and it's sticking to itself. And then there's also this, we're talking about this next week on Sunday. I'll talk about this in terms of water potential. But when water evaporates from the stomata of each of the leaf, it creates a negative pressure potential. And so we'll get into water potential later. But again, the reason why we have this capillary action. So in efforts to make sure we move on to macromolecules. Surface tension is another big one. Our biological example here is, organisms higher altitudes. Pressure walk across water, which is actually going to lower the your ionization of materials. So the water being that universal solvent. So this is important for like just natural. Again, this kind of gets into chemistry a little bit. So things can dissolve. I'm just going to keep pushing along. If you need to take a second in a cell, and remember the cytoplasm is mostly water. So when we talk about water, we're not talking about a pure aqueous. Take a second. We're kind of just past the halfway point. We're talking about a solution that has some water in it. Okay. And if there are water molecules there, your shoulders out, do what you got to do. Partial positives and partial negatives that can disrupt bonds. It's important to also, if you need to close your eyes for a second. We'll get to that later in the lesson today too, about water's ability to disrupt bonds. Okay, we're going to talk about building and breaking molecules. I like this picture right here because it kind of shows this crystallized lattice structure. Most of you have taken chemistry. If you haven't, don't worry about it too much. Floats. Biggest things you need to know are what are present. Just checking the chats, making sure we're okay. The molecules that we're going to be talking about in biology, right? So the big ones that you need to know are Ellie, thank you so much. Carbon, nitrogen, oxygen, I understand why water is cohesive, but why is it sometimes adhesive? Sulfur, right? Sulfur is not present in every single one, so water is going to adhere to other things. Some teachers like to that use are like an acronym, so CHNOPS or CHOMPS. I'm gonna whatever works for you. Actually, when I was in college, they only talked about carbon, oxygen, nitrogen, and hydrogen. You're gonna see my face and for a second. We called it CONHA. When you are in really also class, and this is not a perfect class, I wish it was clear all the way, but hey, I'm plugging my phosphorus is in, our phospholipids. Anyways, when you're in chemistry class and you are told to get the volume of water, that is all I'm gonna talk about for and you get that meniscus most of you have taken chemistry.
You get this like
and honestly,
lower dip, and
as we go through these macromolecules,
like it's creeping up the walls of the water. It's doing that because most glass is made up of
we can all do
silicon
this.
dioxide.
We are nearing,
and
that's actually a polar molecule.
So
when you have
water
similar to how I just
other substances,
at you,
it's because they are polar as well.
Properties of water.
And that's actually a really
same
good
thing for macromolecules because this
that's a really good
that is
application
difficult
question.
to understand, right? That like we have protein, carbs, nucleic acids, and lipids.
Okay, I'm going back to the screen.
All of you can understand that, and you will know it. And you already know it. We have a couple questions about.
I'll leave this here for you. You have this.
I want to direct your attention
you
to
should
this
know
picture
the
right here
that are different from.
If you're a
each
water
one,
molecule
right?
that's
so
not
if you
at
look
the
at what's
surface,
similar, they all have
you
carbon,
are having
hydrogen
oxygen
bond
so if you're
interactions
given information
with water
that
molecules
just talks about
on top of
carbon,
you,
hydrogen,
left, right,
oxygen,
bottom.
Okay, you've got all
and
these
that
interactions
is it.
happening, right? So almost
And
imagine it as like carbs and
you're
lipids
being
both
pulled
have
in
carbon,
four
hydrogen,
different
and
directions.
oxygen, but you should know
the difference between those two.
Water
So like
molecules
carbohydrates are
at
going
the
to have
very
a lot
top
more oxygens.
because in your sugar range,
Don't
right,
have
think
that
about like the
same,
basic
pulling in all directions
interactions. And
nomenclature
so what happens
for
is
sugar
you think
is
about this top layer of water molecules right
so there's
here.
as many oxygens
Because
as
nobody
carbons
is
in that
pulling them
example.
from the top.
So with
The
lipids, you don't
tension
have
they
as many
experience with the
oxygens.
waters to
So
their left
being
and to
aware
their
of those
right
relative
can
amounts
actually
is also
be pretty
helpful.
stabilized. It's almost like pulling a rope.
This is what
right?
you should
So like
be
if you're playing
focusing
tug of
on
war
when you're learning
and someone on the other team isn't holding it very tight and it gets kind of loose, right? It's going to be kind of wobbly.
Huge theme in AP Bio, and what I
disorganized,
mean by this
right?
is you can ask
that
yourself,
very top
how does
layer
the
is like
structure
your
of
rope,
blank
right? And that's why it's called
impact
tension.
its
If any of
function?
you have ever taken physics,
If you can answer
tension
that
is like
about
the force
a
in
lot
rope.
of the structures you're
so
learning,
that top layer
you
is
are
going to be a little bit
amazing.
stronger,
okay?
than
So
the
for
bottom.
example, we have
All right, I'm going to keep going because we are
glycogen,
24 minutes in.
If we
I want to
look
make sure
at
we have
those
time
three
for the rest of
structures here,
macromolecules
and like
finish
these
water.
little
All right, we have specific
hexagons.
If you've taken
Are
chemistry, you've learned about that. But it has to do with
could potentially
water's ability
use
where
for,
it takes
cellular
energy
or
change
the
the temperature. So
which
here
one
this
of
would be an
these looks
application
harder
question
to
where we've
break
got liquid
into?
water has a specific heat of
I've got starch,
4.2,
glycogen, and cellulose.
where iron
Which one of those
has
looks like it might be harder to
0.0005,
break apart?
right? If you've ever touched something metal or iron on a hot day, you could burn yourself.
And you
In
can
fact,
type
my
in the
daughter,
chat,
she
just kind of
stepped
think it in your
on
head.
a metal.
But which one of
grate,
these,
so she was
starch,
like 1 years
glycogen,
old,
cellulose,
in the
which
middle
one
of
looks
a
like
sunny
it's
day,
probably harder
she
to
got second-degree burns
to
on her hands
break
and her
bonds?
feet. Okay, it was the worst day of my life, right? And that's because it takes very little energy because of this.
Yay, you're
this
all
specific
amazing.
heat.
You're like
It
cellulose.
takes very little energy
Yeah, absolutely.
to change
Cellulose.
the
Look at
temperature
how
of that
organized.
substance.
It's like someone's perfect.
Look how much bigger
organized,
liquid water is,
you know,
compared to 0.0005.
clothing drawer, right? Like you've got everything
It is a
organized,
high
everything's
specific
folded
heat. So
perfectly.
to
I'm not gonna
change
wanna
it,
mess
it
that
takes
up.
a
It looks
ton
so
of energy.
great. Where
And that's
starch,
good
it's
for
kind of a hot mess. There's
places
branches
near
everywhere,
water,
right?
Temperature is a
like
little
that
more
for a
regulated. Like
reason,
if
right?
you
That
live near the
structure
ocean,
helps that
right?
function because you
Temperatures
can
of the
easily break
region tend to be
into
more stable
our,
because you're near water. The
you can
other
break and
important
grab
part,
those glucose
because, we care
molecules.
about people and everything,
They
but
still need enzymes.
I'm savvy to the Great
same
Lakes
thing
because
with
that's where I
glycogen,
live. But
those are going to
think
be
about
more
all
easily broken
the
down.
life. We're in biology,
Cellulose
right?
is
We
the
care
stuff
about
that's
living
found in
things.
plant fibers.
So
all
we
the
actually
life
don't
that's
have
living
in
in
this
this beta
water.
amylase. We don't
If
have
the
that,
temperature
so
of
we
water
aren't
could
able to break down
fluctuate
cellulose.
just
So
in a
your
given
doctor
day,
tells you to eat
right?
more
Like in the
fiber.
summer,
Fiber is actually
the
what
air
helps
temperature
kind of
could be as
scoot
low
things
as
along in your large
50, and then in the
intestine.
middle of the day, it could be near 100. And so with that temperature fluctuation, if water could go willy-nilly and get hot, cold, those organisms would suffer, right? Most of them are thermoconformers, which means they become the same temperature as their environment. They're not thermoregulators like we are, right? They don't use their metabolism or they don't, can't like run around and try to generate heat like we do. So they conform to the temperature of their environment. So for living things, it's really important, and it's wonderful that water has high specific heat, that it's not increasing or decreasing in temperature. High heat of vaporization has to do with the fact that it takes a lot of energy as well for water to evaporate. And so biologically speaking, the big example there is evaporative cooling. And again, these aren't things for you to memorize. These are things for you to just recognize as examples of these different properties. So you might be given an example of something totally different than what I've given you today. But hopefully you can reason through which property it is they're talking about and then describe how water's polarity, its hydrogen binding, its polarity, and its structure are attributed to that property. Just asking about, carbon, nitrogen, oxygen, hydrogen, it's protein. Okay, how are macromolecules built and broken? This is something that is always happening, and it's one of those things where I'll teach this unit 1, and then I'll forget to continue talking about it because it's just an assumed thing, but continue talking about it, okay? So when we talk about monomers becoming polymers, and we mentioned dehydration synthesis, right? And so how we dehydrate, we remove a hydroxyl and a hydrogen a hydroxyl from one monomer and a hydrogen from another monomer. We take out water, and that links your two monomers together in dehydration synthesis. This is happening all the time throughout the rest of our units. Or if we're talking about, hey, like we cleaved this bond, or this enzyme does this, water's playing a role there too. So don't think of this as just, this is something to learn about macromolecules. Think about it, it's building and breaking bonds. It's not just for macromolecules. And then hydrolysis is the opposite of dehydration synthesis, hydrolysis, so water lysis, which means we lyse or we cut with water. If any of you are super into Latin, I always tell my students, I used to give them a root word packet at the beginning of the year. I used to teach anatomy too, and I'd make them memorize all the root words. And then all of a sudden we'd be learning stuff and they'd be like, I know what that is, before I even taught it. So they were able to kind of deduce the language and the vocabulary by just knowing root words. So anyway, and not that I want to give you more to do. I know you are all very, very busy. But you should know these two processes, right? We dehydrate. Some teachers call it condensation for dehydration synthesis, but it is known as the AP curriculum framework calls it dehydration synthesis. And then hydrolysis, we are breaking bonds. Water is breaking those bonds. Okay, I have a couple slides that goes over each macromolecule. So for each slide, I'm going to give you about 30 seconds to read it. All right, so with carbohydrates, something that's not written down, again, is that structure function. And a huge reason why carbohydrates vary in their structure, right? Whether you have branches that are kind of all over the place, or if you have very clean linear branches or connections, has to do with the actual monomer. All right, so if you look at this monomer right here, right, the inversions of whether or not you have like a hydroxyl down here or up here, or if you have a hydroxyl up or down, those are different isomers that can be present, and they're just different versions of your glucose. So if you've ever heard of the difference between like fructose or lactose or glucose, right? Those are all varying structures. Yes, they're still sugars, right? So big picture, they're sugars, just like how I can talk about how I love soda or pop, wherever you're from, sorry, but you know, pop, right? A soft drink. If I love pop and I just ask someone to get me a pop and they bring me an orange pop, I hate orange pop and I'd be really mad at them, but I didn't tell them the specific pop I wanted, right? So with carbohydrates, the very specific monomer, whether it's fructose, lactose, or even with glucose, we have alpha glucose, we have beta glucose, and I think I have a picture of that. Hold on. Here it is. So there's alpha glucose, there's beta glucose, and depending on the actual type of your sugar monomer is what allows those different structures to happen. Okay, so here's lactose, here's maltose. This is two maltoses together, two lactoses together. Notice how with the sucrose, the oxygen's down here, right? They are different from one another. When you take organic chemistry, you will learn how to name all these different things, learn why it's alpha versus beta. You'll learn about all sorts of different, molecular things that you do not need to know for this class. But you should be able to talk backwards. And if you get a question that asks you, explain how those, why cellulose has a different structure than starch, you'd want to go back to the monomer and say those branches or linear connections started from different monomers or different versions of a monomer, right? So we have still glucose, still sugar, but it looks different, and so it's going to have different linkages. Cellulose has beta linkages, starch has alpha linkages, and so that's why we don't have beta amylase, so we cannot break those beta bonds. All right, lipids, take 30 seconds to read. Just for those of you who are not reading the chat, you do not need to know the differences in these structures, no. So you will not need to know does, cellulose have beta glucose or alpha glucose? You do not need to know that. But you should know that if the structure of the monomer is different, then the polymer will be shaped different. All right? Yes. All right, let's keep going. Lipids. So lipids are normally, they're nonpolar, right? They have lots of carbons and hydrogens, right? So just like that methyl group, they've got carbons and hydrogens. Phospholipids are a little bit different, right? They're amphipathic. So they have a hydrophobic section because of that phosphate group with those highly electronegative oxygens. And then they have a I think I said hydrophobic heads. I didn't mean to say that. Hydrophilic heads. And then you have these hydrophobic tails. So they're not going to want water to hang around. They're not going to want to interact with water. And water's not going to interact with it either because it's not polar. Lipids can be used for all sorts of things. Insulation. They're in your cell membranes. And here's something. I was actually talking to a student today. And we were talking about the mitochondrial membrane. And a lot of people don't realize that the inner membrane of the mitochondria is also made up of phospholipids. We so often just draw it as a squiggly line, but it's a membrane. It's an actual membrane, right? That where the electron transport chain happens. Same thing as the thylakoid. It's a membrane, right? It's made up of phospholipids. So they're more applications than just lipids and it's here for hormones and fat storage and things like that. But it's cell membranes, and it's the inner membranes of these membrane-bound organelles. So when you learned about membrane-bound organelles next week, they have phospholipids in them. Okay, proteins, these are our most diverse macromolecule. They can be anything. I always tell my students, when in doubt, you're looking at a protein most likely. Also, the biggest thing that students forget when we talk about enzymes, remember that enzymes are a protein, okay? This is where you're going to see that sulfur potentially. The sequence matters. So when we talk about amino acids that are the building block of proteins, the sequence that those amino acids come together matters a whole lot for its structure. Just like how, and again, look at these commonalities. These are things for you to be able to discuss. The sequence of amino acid matters. The type of monosaccharide matters. You don't need to memorize it, but you need to know. You don't need to memorize the monosaccharide, but you need to know that, like, hey, look at that similarity there. That like the type of monosaccharide allows the polysaccharide or the macromolecule to look a certain way and function a certain way. And hey, so does the amino acid, right? We talk about protein folding, and we'll get more into this in our protein synthesis unit. But your amino acids, the sequence matters because in primary structure is just the peptide bonds, right? You get peptide bonds between your amino acids, just a covalent bond. The amino acids then start to interact with one another. You get local folding, so that's just hydrogen bonds between your carboxyl and amine areas of the amino acids. Tertiary structure, you build on that, right? And again, this is all because of that specific sequence that this folding happens. Tertiary structure is where you have your side chain interactions. So that's your R groups. So you might have like if you have a charged amino acid, you might have your positives and negatives hanging out next to each other. You might have your hydrophobic amino acids all hiding. You have your hydrophilic on the outside of your protein. So this is where you're starting to get a more functional protein. Quaternary structure is when you have more than one polypeptide together. So more than one primary structure that has been folded. So like hemoglobin has 4 subunits. So you would have 4 primary protein structures that then have to all fold and come together. Okay, I'm going to stop looking at the chat because I have to fly in these next few slides. If you're interested in how perms happen, go ahead and Google it. It's good research. It's actually really good reading to learn about that local folding and something called the disulfide bond, which is present in some proteins, especially your hair. Okay, looking at amino acids, you should, at this point, with your skills of polarity, be able to look at something and talk about things that are hydrophobic, right? So like all of these here in green are hydrophobic because it's just carbons, hydrogens, and rings. If you see things with the side chains having lots of oxygens, you're getting into something polar. Sometimes they'll even be nice and show you an amino acid that has positives and negatives, and then it's definitely not hydrophobic. So I'm gonna keep clicking. All right, concept check, friend. Okay, let's end the 5 minutes with our nucleic acids, our final macromolecule. By now, you probably know so much about nucleic acids because there's a unit where you're learning all about them. You're learning about DNA replication, then you're learning about protein synthesis. Then it even comes up again later when you talk about evolution and natural selection, right? So I feel like nucleic acids, most students are wonderful at. But just in case you want a refresher, be sure you're able to talk about this nucleotide. You do not need to draw it, but be aware that a nucleotide has 3 things: phosphate, a sugar, and a base. Okay? Be aware of that. Also be aware of what makes up the backbone and what makes up the bridges. So know that it's your bases that bind and then your phosphate sugar that make up the backbone of your DNA. Other big things to make sure you know is that your phosphate sugar backbone, that is a covalent bond. So phosphate sugar are very strong, which makes sense because you don't want water to have like disrupted you don't want DNA, your heredity information, to have bonds that just break apart because some enzyme came around. But the hydrogen bonds are more easily disrupted, which is a good thing because then we can have something like helicase come in and unzip the DNA when we need to replicate it. So make sure you know where you can find covalent bonds, where you can find hydrogen bonds. DNA base pairing, this is based off of Chargaff's rules. So if I told you, hey, I've got this DNA strand, this molecule, and I've got 14% cytosine, go ahead and try to deduce what percentages all the other ones are. This is a very, you've probably seen a question just like this, but it's always good to get practice because you'll always have someone who does a silly math problem or math mistake and they're like, I can't believe I did that. Take a second, 14% cytosine. How much are you going to find elsewhere? You are all wonderful. Great job. Good. So I kind of tried to trip you up a little bit and by putting 14% G in the middle here. But yeah, if you know G is, if you know C is 14%, then G will be 14. And then it's just math of, you know, that 72% needs to be shared between thymine and adenine. If you have any questions about this, please, you know, stay after and continue the conversation on Discord. People will be there to help. I can't promise that I'll be there since I have two babies, not two babies, but two big kids that need to go to bed. All right, just like protein, DNA has directionality. So we've got a 5 to 3' side and a 3 to 5'. This is something you learned when you very first learned about DNA and the fact that it's this anti-parallel strand. They don't run parallel, they run anti-parallel. So I always envision like the phosphate and the sugar as being like a person standing up. Like I think of the phosphate as a person with a head, right? And so on one end of the strand, your 5' end is the person's head standing. And then your other strand, it's someone doing a handstand. So they're upside down. I don't know. It's just my weird way of remembering it, that they're literally upside down. And so your 5' is always going to be the carbon attached to the phosphate group. And your 3' is always going to be the carbon that's attached to your sugar right here. Okay, so it has directionality. I'm just kind of showing you. Those are my hand-drawn images, aren't they funny? I encourage you to draw yourself for yourself. Here's just a picture of how 5 to 3' addition works. And I know that's not until later units, but I just wanted to kind of give you a nice visualization of when we say it is 5 to 3' and we add nucleotides, it's literally the 5' end gets added to the 3' end. Okay, so 5' of one nucleotide gets added to the 3' end. And that goes on and on and on until the whole thing is replicated. Last concept question of tonight, what type of bond is formed when the phosphate group attaches to the deoxyribose sugar ring? Oops, sorry. It is a covalent bond. Yay!